Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Endoplasmic reticulum. 79 languages. Cell biology. Animal cell diagram. Components of a typical animal cell. 1 nucleolus. 2 nucleus. 3 ribosome, dots as part of 5. 4 vesicle. 5 rough endoplasmic reticulum. 6 Golgi apparatus, or Golgi body. 7 cytoskeleton. 8 smooth endoplasmic reticulum. 9 mitochondrion. 10 vacuole. 11 cytosol, fluid that contains organelles, with which comprises cytoplasm. 12 lysosome. 13 centrosome. 14 cell membrane. The endoplasmic reticulum, is a part of a transportation system of the eukaryotic cell, and has many other important functions such as protein folding. It is a type of organelle made up of 2 subunits: rough endoplasmic reticulum, RER, and smooth endoplasmic reticulum, SER. The endoplasmic reticulum is found in most eukaryotic cells and forms an interconnected network of flattened, membrane enclosed sacs known as cisternae, in the RER, and tubular structures in the SER. The membranes of the are continuous with the outer nuclear membrane. The endoplasmic reticulum is not found in red blood cells, or spermatozoa. The 2 types of share many of the same proteins, and engage in certain common activities such as the synthesis of certain lipids and cholesterol. Different types of cells contain different ratios of the 2 types of depending on the activities of the cell. RER is found mainly toward the nucleus of cell and SER towards the cell membrane or plasma membrane of cell. The outer, cytosolic, face of the RER is studded with ribosomes that are the sites of protein synthesis. The RER is especially prominent in cells such as hepatocytes. The SER lacks ribosomes and functions in lipid synthesis but not metabolism, the production of steroid hormones, and detoxification. 1. The SER is especially abundant in mammalian liver and gonad cells. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Micrograph of rough endoplasmic reticulum. Network around the nucleus, shown in the lower right-hand area of the picture. Dark small circles in the network are mitochondria. 1 nucleus, 2 nuclear pore, 3 rough endoplasmic reticulum, RER, 4 smooth endoplasmic reticulum, SER, 5 ribosome on the rougher 6 proteins that are transported. 7 transport vesicle, 8 Golgi apparatus, 9 CIS face of the Golgi apparatus, 10 trans face of the Golgi apparatus, 11 cisternae of the Golgi apparatus. The was observed by light microscopy by Garnier in 1897, who coined the term ergastoplasm. 2, 3. The lacy membranes of the endoplasmic reticulum were first seen by electron microscopy in 1945 by Keith R. Porter, Albert Claude, and Ernest F. Fullam. 4. Later, the word reticulum, which means network, was applied by Porter in 1953 to describe this fabric of membranes. 5. Structure, edit. The general structure of the endoplasmic reticulum is a network of membranes called cisternae. These sac-like structures are held together by the cytoskeleton. The phospholipid membrane encloses the cisternal space, or lumen, which is continuous with the perinuclear space but separate from the cytosol. The functions of the endoplasmic reticulum can be summarized as the synthesis and export of proteins and membrane lipids, but varies between and cell type and cell function. The quantity of both rough and smooth endoplasmic reticulum in a cell can slowly interchange from one type to the other, depending on the changing metabolic activities of the cell. Transformation can include embedding of new proteins in membrane as well as structural changes. Changes in protein content may occur without noticeable structural changes. 6. 7. Rough endoplasmic reticulum, edit. The surface of the rough endoplasmic reticulum, often abbreviated RER or rougher, also called granular endoplasmic reticulum, is studded with protein manufacturing ribosomes, giving it a rough appearance, hence its name. 8. The binding site of the ribosome on the rough endoplasmic reticulum is the translocon. 9. However, the ribosomes are not a stable part of this organelle's structure, as they are constantly being bound and released from the membrane. A ribosome only binds to the RER once a specific protein nucleic acid complex forms in the cytosol. This special explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF 3D rendering of endoplasmic reticulum. A 2-minute animation showing how a protein destined for the secretory pathway is synthesized and secreted into the rough endoplasmic reticulum, which appears at the upper right. Approximately halfway through the animation, complex forms when a free ribosome begins translating the mRNA of a protein destined for the secretory pathway. 10. The first 530 amino acids polymerized encode a signal peptid, a molecular message that is recognized and bound by a signal recognition particle, SRP. Translation pauses and the ribosome complex binds to the RER translocon, where translation continues with the nascent, new, protein forming into the RER lumen and / membrane. The protein is processed in the lumen by an enzyme, a signal peptidase, which removes the signal peptid. Ribosomes at this point may be released back into the cytosol. However, non-translating ribosomes are also known to stay associated with translocons. 11. The membrane of the rough endoplasmic reticulum is in the form of large double membrane sheets that are located near and continuous with the outer layer of the nuclear envelope. 12. The double membrane sheets are stacked and connected through several right or left-handed helical ramps, the Tarosaki ramps, giving rise to a structure resembling a parking garage. 13. 14. Although there is no continuous membrane between the endoplasmic reticulum and the Golgi apparatus, membrane-bound transport vesicles shuttle proteins between these two compartments. 15. Vesicles are surrounded by coating proteins called copy and copii. Copy targets vesicles to the Golgi apparatus and copy marks them to be brought Back to the rough endoplasmic reticulum. The rough endoplasmic reticulum works in concert with the Golgi complex to target new proteins to their proper destinations. The second method of transport out of the endoplasmic reticulum involves areas called membrane contact sites, where the membranes of the endoplasmic reticulum and other organelles are held closely together, allowing the transfer of lipids and other small molecules. 17. The rough endoplasmic reticulum is key in multiple functions. Manufacture of lysosomal enzymes with a mannose 6-phosphate marker added in the CIS Golgi network. 18. Manufacture of secreted proteins, either secreted constitutively with no tag or secreted in a regulatory manner involving clathrin and paired basic amino acids in the signal peptide. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Electron micrograph showing smoother arrow in mouse tissue at 110,510 magnification. Integral membrane proteins that stay embedded in the membrane as vesicles exit and bind to new membranes. RAB proteins are key in targeting the membrane. SNAP and SNARE proteins are key in the fusion event. Initial glycosylation as assembly continues. This is N-linked. O-linking occurs in the Golgi. N-linked glycosylation, if the protein is properly folded, oligosaccharidyl recognizes the AA sequence NXS or NXT, with the S/T residue phosphorylated, and adds a 14 sugar backbone, 2N-acetylglucosamine, 9 branching mannose, and 3 glucose at the end, to the side chain nitrogen of ASN. Smooth endoplasmic reticulum, edit. In most cells the smooth endoplasmic reticulum, abbreviated SER, is scarce. Instead there are areas where the is partly smooth and partly rough. This area is called the transitional The transitional gets its name because it contains exit sites. These are areas where the transport vesicles which contain lipids and proteins made in the detach from the and start moving to the Golgi apparatus. Specialized cells can have a lot of smooth endoplasmic reticulum, and in these cells the smoother has many functions. 6. It synthesizes lipids, phospholipids, 19, 20, 21, and steroids. Cells which secrete these products, such as those in the testes, ovaries, and sebaceous glands, have an abundance of smooth endoplasmic reticulum. 22. It also carries out the metabolism of carbohydrates, detoxification of natural metabolism products and of alcohol and drugs, attachment of receptors on cell membrane proteins, and steroid metabolism. 23. In muscle cells, it regulates calcium ion concentration. Smooth endoplasmic reticulum is found in a variety of cell types, both animal and plant, and it serves different functions in each. The smooth endoplasmic reticulum also contains the enzyme glucose 6-phosphatase, which converts glucose 6-phosphate to glucose, a step in gluconeogenesis. It is connected to the nuclear envelope and consists of tubules that are located near the cell periphery. These tubes sometimes branch, forming a network that is reticular in appearance. 12. In some cells, there are dilated areas like the sacs of rough endoplasmic reticulum. The network of smooth endoplasmic reticulum allows for an increased surface area to be devoted to the action or storage of key enzymes and the products of these enzymes. Citation needed. Sarcoplasmic reticulum, edit. Main article, Sarcoplasmic reticulum. See also, calcium-induced calcium release. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Skeletal muscle fiber, with sarcoplasmic reticulum colored in blue. The sarcoplasmic reticulum, SR, from the Greek sarx, flesh, is smoother found in muscle cells. The only structural difference between this organelle and the smooth endoplasmic reticulum is the composition of proteins they have, both bound to their membranes and drifting within the confines of their lumens. This fundamental difference is indicative of their functions. The endoplasmic reticulum synthesizes molecules, while the sarcoplasmic reticulum stores calcium ions and pumps them out into the sarcoplasm when the muscle fiber is stimulated. 24-25. After their release from the sarcoplasmic reticulum, calcium ions interact with contractile proteins that utilize ATP to shorten the muscle fiber. The sarcoplasmic reticulum plays a major role in excitation-contraction coupling. 26. Functions, edit. The endoplasmic reticulum serves many general functions, including the folding of protein molecules in sacs called cystinae and the transport of synthesized proteins in vesicles to the Golgi apparatus. Rough endoplasmic reticulum is also involved in protein synthesis. Correct folding of newly made proteins is made possible by several endoplasmic reticulum chaperone proteins, including protein disulfide isomerase, PDI, ERP29, the HSP70 family member BIP/GRP78, calnexin, calreticulin, and the peptidyl prylyl isomerase family. Only properly folded proteins are transported from the rougher to the Golgi apparatus. Unfolded proteins cause an unfolded protein response, as a stress response in the disturbances in redox regulation, calcium regulation, glucose deprivation, and viral infection. 27, or the overexpression of proteins. 28, can lead to endoplasmic reticulum stress response, stress, a state in which the folding of proteins slows, leading to an increase in unfolded proteins. This stress is emerging as a potential cause of damage in hypoxia/ischemia, insulin resistance, and other disorders. 29. Protein transport, edit. Secretory proteins, mostly glycoproteins, are moved across the endoplasmic reticulum membrane. Proteins that are transported by the endoplasmic reticulum throughout the cell are marked with an address tag called a signal sequence. The N-terminus, one end, of a polypeptide chain, i.e., a protein, contains a few amino acids that work as an address tag, which are removed when the polypeptide reaches its destination. Nascent peptides reach the via the translocon, a membrane-embedded multiprotein complex. Proteins that are destined for places outside the endoplasmic reticulum are packed into transport vesicles and moved along the cytoskeleton toward their destination. In human fibroblasts, the is always co-distributed with microtubules and the Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Ca2+ antagonized transport into the endoplasmic reticulum. Cartier model. Depolymerization of the latter causes its co-aggregation with mitochondria, which are also associated with the 30. The endoplasmic reticulum is also part of a protein sorting pathway. It is, in essence, the transportation system of the eukaryotic cell. The majority of its resident proteins are retained within it through a retention motif. This motif is composed of 4 amino acids at the end of the protein sequence. The most common retention sequences are CTL for lumen-located proteins and KKXX for transmembrane proteins. 31. However, variations of CTL and KKXX do occur, and other sequences can also give rise to endoplasmic reticulum retention. It is not known whether such variation can lead to sublocalizations. There are 3 CTL, 1, 2 and 3, receptors in mammalian cells, and they have a very high degree of sequence identity. The functional differences between these receptors remain to be established. 32. Bioenergetics regulation of ATP supply by a Cartier mechanism, edit. The endoplasmic reticulum does not harbor an ATP regeneration machinery, and therefore requires ATP import from mitochondria. The imported ATP is vital for the to carry out its housekeeping cellular functions, such as for protein folding and trafficking. 33. The ATP transporter, SLC35B1/AXA, was recently cloned and characterized. 34, and the mitochondria supply ATP to the through a Ca2+ antagonized transport into the Cartier mechanism. 35. The Cartier mechanism shows sensitivity to cytosolic Ca2+ ranging from high Nm to low M range, with the Ca2+ sensing element yet to be identified and validated. 36. Clinical significance, edit. Increased and supraphysiological stress in pancreatic beta cells disrupts normal insulin secretion, leading to hyperinsulinemia. 37, and consequently peripheral insulin resistance associated with obesity in humans. 38. Human clinical trials also suggested a causal link between obesity-induced increase in insulin secretion and peripheral insulin resistance. 39. Abnormalities in XBP1 lead to a heightened endoplasmic reticulum stress response and subsequently causes a higher susceptibility for inflammatory processes that may even contribute to Alzheimer's disease. 40. In the colon, XBP1 anomalies have been linked to the inflammatory bowel diseases including Crohn's disease. 41. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The unfolded protein response, UPR, is a cellular stress response related to the endoplasmic reticulum. 42. The UPR is activated in response to an accumulation of unfolded or misfolded proteins in the lumen of the endoplasmic reticulum. The UPR functions to restore normal function of the cell by halting protein translation, degrading misfolded proteins, and activating the signaling pathways that lead to increasing the production of molecular chaperones involved in protein folding. Sustained overactivation of the UPR has been implicated in prion diseases as well as several other neurodegenerative diseases, and the inhibition of the UPR could become a treatment for those diseases. 43. See also, edit. Ribosome-associated vesicle. References, edit. 1 carat endoplasmic reticulum, rough and smooth. British Society of Cell Biology. Archived from the original on the 24th of November, 2015. Retrieved the 21st of November, 2015. 2 carat Garnier, c. 1897. Les filaments basaux des cellules glandulaires. Note préliminaire. Bibliographie anatomique. 5. 278-289. OCLC 4934411682. 3 carat Buvat, A. 1963. Electron Microscopy of Plant Protoplasm. International Review of Cytology. Volume 14. Volume 14. pp. 41155. Do. 10.1016/S0074-7696, 08, 60021-2. ISBN 978-0-12-364314-8. PMID 14283576. 4 carat Porter K.R., Claude, Fulham F., March 1945. A Study of Tissue Culture Cells by Electron Microscopy. Methods and Preliminary Observations. The Journal of Experimental Medicine. 81, 323346. Do. 10.1084/gem.81.3.233. PMC 21359493. PMID 19871454. 5 carat Porter K.R., May 1953. Observations on a submicroscopic basophilic component of cytoplasm. The Journal of Experimental Medicine. 97, 572752, 10.1084/gem.97.5.727. PMC 213695. PMID 13052830. 6 carat AB Alberts B., Johnson O., Lewis J., Raff M., Roberts K., Walter P., 2002. Molecular Biology of the Cell. 4th ed. New York, Garland Science. ISBN 978-0-8153-3218-3. Archived from the original on 2017. 10-03. 7 carat Cooper G.M., 2000. The Cell, A Molecular Approach, 2nd ed. Washington, DCASM Press. ISBN 978-0-87893-106-4. 8 carat Reticulum. The Free Dictionary. 9 carat Gorlick D., Pren S., Hartman E., Kalizku, Rappaport Ta., October 1992. A mammalian homologous. Sec 61P and SECYP is associated with ribosomes and nascent polypeptids during translocation. Cell. 71, 3489503 do. 10.1016/0092-8674, 92, 90517G. PMID 14233609. S2 SID 19078317. 10 carat Lodish H., it al. 2003. Molecular Cell Biology, 5th ed. W. H. Freeman. pp. 659666. ISBN 978. 0-7167-4366-8. 11 carat Sizer RM, Nikita CV, October 2000. The fate of membrane-bound ribosomes following the termination of protein synthesis. The Journal of Biological Chemistry. 275, 43338207. Do. 10.1074/jbc.m0044622200. PMID 10931837. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 12 AB Scheibe to Y, Voelz GK, Rappaport Ta, August 2006. Rough sheets and smooth tubules. Cell 126, 34359 do. 10.1016/j.cell.2006.07.019. PMID 16901774. S2 SID 16107069. 13 Tarozaki M, SHEMESHT, Casthuri N, Clem RW, Skalek R, Hayworth KJ, Hand R, Yankova M. Huber G, Leichtman JW, Rappaport Ta, Kozlov mm, July 2013. Stacked endoplasmic reticulum sheets are connected by helicoidal membrane motifs. Cell 154, 228596. Do. 10.1016/j.cell.2013.06.031. PMC 3767119. PMID 2387020. 14 Guven J, Huber G, Valencia DM, October 2014. Tarozaki spiral ramps in the rough endoplasmic reticulum. Physical Review Letters 113, 18 188101. Bib Code 2014 PHRVL.113R8101G. Do. 10.1013/PHYSRevlet.113.1888101. PMID 253963996. 15 Endoplasmic reticulum. ND. McGraw-Hill Encyclopedia of Science and Technology. Retrieved 13 September 2006, from Answers.com website. Answers, the most trusted place for answering life's questions. Answers.com. Archived from the original on 16 November 2006. Retrieved 13 September 2006. 16 Levine T, September 2004. Short-range intracellular trafficking of small molecules across endoplasmic reticulum junctions. Trends in Cell Biology. 14, 948390. Do. 10.1016/j.tcb.2004.07.017. PMID 15350976. 17 Levine T, Low UNC, August 2006. Inter-organelle membrane contact sites, through a glass, darkly. Current opinion in cell biology. 18, 43718 10.1016/j.seb.2006.06.011. PMID 16806880. 18 Aval, Tomislav, Ju, Jing, Tian, Weiwa, Remelwal, San, Yang, Zhang, Clausen, Henrik. Heck, Albert Jr. 1 January 2019. Targeted analysis of lysosomal directed proteins and their sites of Manners 6-phosphate modification *S. Molecular and cellular proteomics. 18, 11627. Do. 10.1074/MCP.RA118.0000967. ISSN 1535-9476. PMC 6317476. PMID 3023720. 19-carat Canon, Matthew Kumar, Lahiri, Suyoy, Lu, Lee Ka, Chowdhury, Vineet, Prinz, William O. March 2017. Phosphatidylserine synthesis at membrane contact sites promotes its transport out of the Journal of Lipid Research. 58, 3553562 do. 10.1094/JLR.M072959. PMC 5335585. PMID 28119445. 20-carat Canon, Matthew Kumar, Riek Hof, Wayne R. Voelker, Dennis R. February 2015. Transport of phosphatidylserine from the endoplasmic reticulum to the site of phosphatidylserine decarboxylase 2. In yeast, phosphatidylserine transport to the locus of PSD2P. Traffic 16, 21233134. Do. 10.1111/TRA.12236. PMID 253555612. S2 SID 34302. 21-carat Friedman, Jonathan R. Cannon, Matthew Kumar, Tulme, Alexandre, Jan, Calvin H. Weissman. Jonathan S. Prinz, William A. Nunnery, Jody, January 2018. Lipid homeostasis is maintained by dual targeting of the mitochondrial PAE biosynthesis enzyme to the developmental cell. 44, 2261. 270.E6. Do. 10.1016/J.Devsel.2017.11.023. PMC 5975648. PMID 292905883. 22-carat functions of smoother. University of Minnesota Duluth. 23-carat Maxfield Fr. Vestner D. October 2002. Intracellular cholesterol transport. The Journal of Clinical Investigation. 110, 78918 do. 10.1172/JCI16500. PMC 151159. PMID 1237024. 24-carat Toyoshima C., Nakasako M., Nomura H., Ogawa H., June 2000. Crystal structure of the calcium pump of sarcoplasmic reticulum at 2.6 of resolution. Nature. 405, 6787 64755. Bib code 2000 nature.405647t. Do. 10.1038/350150017. PMID 10864315. S2 SID 4316039. 25-carat Goodman SR, the 26th of November 2007. Medical Cell Biology, 3rd ed. Academic Press. P69. ISBN 9780080919317. 26-carat Martini F., Nath J., Bartholomew E., 2014. Fundamentals of Anatomy and Physiology, 10th ed. Pearson. ISBN 978-0321909077. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 27 Zoo C, Bale Lemaitre B, Reed J.C., October 2005. Endoplasmic reticulum stress, cell life and death. Decisions. The Journal of Clinical Investigation. 115. 10 2656 64 do 10.1172 JCI 26373 PMC 1236697 PMID 162000199 28 Kobia L. Z.C. Bode J. October 2012. Development of a novel stress-based selection system for the isolation of highly productive clones. Biotechnology and Bioengineering. 109, 10 2599 611. Do 10.1002/bit.24527. PMID 2251090. S2 SID 258582120. 29 Oscan U, Cao Q, Yilmas E, Li R, Iwakoshi NN, Osdalan Tunkman G, Gorgun C, Glimcher LH. Hotamizlagil GS, October 2004. Endoplasmic reticulum stress links obesity, insulin action, and type 2 diabetes. Science. 306, 5695 45761. Bibcode 2004 SCI 3064570. Do 10.1126/science.11031660. PMID 154869293. S2 SID 225173995. 30 Soltis BJ, Gupta RS, 1992. Interrelationships of endoplasmic reticulum, mitochondria, intermediate filaments, and microtubules, a quadruple fluorescence labeling study. Biochemistry and Cell Biology. 70, 1011 1174 86 do 10.1139/092-163. PMID 1363623. 31 Storniulo M, Lotti LV, Borgi Sen, Torisai Mr., Motola G, Martia G, Bonatti S, March 2003. CTL and KKXX retrieval signals appended to the same reporter protein determine different trafficking between endoplasmic reticulum, intermediate compartment, and Golgi complex. Molecular biology of the cell. 14, 388990 do 10.1091/mbc.e02-08-0468. PMC 151567. PMID 126311711. 32 Rakel I, Alan and H, Salo K, Jurvansua J, Nguyen VD, Latvaranta M, Ruddick L, December 2007. A molecular specificity code for the 3 mammalian CTL receptors. The Journal of Cell Biology. 179, 61193 204 do 10.1083/JCB.2007051880. PMC 214024. PMID 180869116. 33 Claremont, CA, de Mayo, A, Hirschberg, CB, the 25th of February 1992. Translocation of ATP into the lumen of rough endoplasmic reticulum-derived vesicles and its binding to luminal proteins including BIP, GRP 78, and GRP 94. The Journal of Biological Chemistry. 267, 3983 10.1016/S0021. 9258, 19, 50622-6. PMID 1740446. 34 Klein, Marie Christine, Zimmermann, Katharina, Sko, Stefan, Landini, Martina, Clemens, Patrico. W. Altensel, Jacqueline, Jung, Martin, Krauser, Elmar, Nguyen, Dewey, Helms, Volkhard, Rettig, Jens. Feschertrist, Claudia, Cavalli, Adolfo, Hoth, Marcus, Bogeski, Ivan, Neuhaus, H. Eckehardt. Zimmermann, Richard, Lang, Sven, Haferkamp, Ilka, the 28th of August 2018. AXA is an ATP/ADP exchanger in the membrane of the endoplasmic reticulum. Nature Communications. 9, 13489. Bibcode 2018 NATCO 9.3489K. Do 10.1038/S41467-018-06003-9. PMC 6113206. PMID 30154480. 35 Yong, Jing, Biscoff, Helmut, Bergstaller, Sandra, Sirin, Marina, Murphy, Anne, Malley, Roland. Kaufman, Randall J., the 9th of September 2019. Mitochondria supply ATP to the through a mechanism antagonized by cytosolic Ca2+. eLife. 8 do 10.7554/eLife.49682. PMC 6763289. PMID 31498082. 36 Yong, Jing, Biscoff, Helmut, Bergstaller, Sandra, Sirin, Marina, Murphy, Anne, Malley, Roland. Kaufman, Randall J., 2019. Mitochondria supply ATP to the through a mechanism antagonized by cytosolic Ca2+. eLife. 8, E49682. Do 10.7554/eLife.49682. ISSN 2050-084X. PMC 6763289. PMID 31498082. 37 Yong, Jing, Johnson, James D. Arvan, Peter, Han, Gisiuk, Kaufman, Randall J. August 2021. Therapeutic opportunities for pancreatic cellular stress in diabetes mellitus. Nature Reviews. Endocrinology. 17, 845547 do 10.1038/S41574-021-00510-4. PMC 8765009. PMID 34163039. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 38 Van Vliet, Stefan, Co, Han Chow E. Patterson, Bruce W. Yoshino, Mayako, La Forest, Richard. Groppler, Robert J. Klein, Samuel, Mittendorfer, Bettina, 1 October 2020. Obesity is associated with increased basal and postprandial cell insulin secretion even in the absence of insulin resistance. Diabetes. 69, 10 2112 2119 do. 10.2337/db20-0377 PMC 7506835 PMID 3265141 39 Mittendorfer, Bettina, Patterson, Bruce W. Smith, Gordon I. Yoshino, Mayako, Klein, Samuel, 1. February 2022. Beta cell function and plasma insulin clearance in people with obesity and different glycemic status. Journal of Clinical Investigation. 132, 3E154068 do. 10.1172/JCI154068 PMC 88033444 PMID 349055513 40 Casas Tinto S, Chang Y, Sanchez Garcia J, Gomez Velázquez M, Rincon Limas D, Fernandez. Funes P, June 2011. The stress factor XBP1S prevents amyloid beta neurotoxicity. Human Molecular Genetics. 2011 2144 60 do. 10.1093/HMG/DDR100 PMC 3090193 PMID 21389082 41 Kayser, Leah, Franco, Glickman J.N., Zeissig S., Tilgage, Nguyen Huis E., Higgins D., Schreiber S., Glimcher L.H., Blumberg R.S., September 2008. XBP1 links stress to intestinal inflammation and confers genetic risk for human inflammatory bowel disease. Cell 134, 574356. Do. 10.1016/J.cell.2008.07.021 PMC 2586148 PMID 1877538 42 Walter, Peter. Peter Walter's short talk, Unfolding the UPR. iBiology. 43 Moreno J.A., Halliday M., Molloy C., Radford H., Verity N., Arkston J.M., Autori C.A., Willis A., Fisher P.M. Barrett The, Malak I.G.R., October 2013. Oral treatment targeting the unfolded protein response. Prevents neurodegeneration and clinical disease in prion-infected mice. Science Translational Medicine. 5, 206-206-Ra138 do. 10.1126/Cytransome.3006767 PMID 2410777 S2 SID 255700626 External links, edit. Endoplasmic reticulum structure, types and functions. Endoplasmic reticulum. Lipid and protein composition of endoplasmic reticulum in OPM database. Animations of the various cell functions referenced here archived the 22nd of April 2008 at the Wayback Machine. Category: Endoplasmic reticulum. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.